Hello everyone, this is lesson number 11 for clinical bacteriology, and in this screencast lecture we're going to discuss gram-negative cocci. So these are our learning objectives for this session. At the end of this lecture, you should be able to number 1, describe the characteristics of the pathogenic Neisseria species in Moraxella catarrhalis. Number 2, explain the importance of proper specimen collection and transport for culture and isolation of pathogenic Neisseria. Number 3, identify the different culture media used for the isolation of Neisseria and Moraxella. Number 4, explain the principles of biochemical tests used to identify Neisseria and Moraxella. And number 5, identify associated diseases caused by your pathogenic Neisseria species and your Moraxella catarrhalis infections. So let's start with the Neisseria species. Neisseria species belong to the family Neisseriaceae. So organisms belonging to the genus Neisseria are obligate aerobes, non-motile, non-hemolytic, fastidious, and capnophilic. So yung mga bacteria na under sa Neisseria na genus, so they are obligate aerobes, meaning they won't survive in the absence of atmospheric oxygen. They're also non-motile, meaning they're non-flagellated. They are non-hemolytic, meaning they do not produce enzymes that destroy or that lyse red blood cells. They are fastidious and capnophilic. So, isa sa mga unique characteristic class ng mga pathogenic Neisseria natin ay they are fastidious organisms. Ibig sabihin ng fastidious, increased ang kanilang nutrient requirements compared to other organisms. That is why they won't grow on routine culture media except on chocolate agar. Diba, in essence, ang ating chocolate agar ay parang blood agar. But the difference is that in chocolate agar, the red blood cells are lysed. Okay? Kasi bakit siya na-lyse? Kasi 'di ba nung inad natin yung blood during the preparation of your chocolate agar, mainit pa yung melted agar natin, therefore causing the lysis of your added red blood cells or your added blood. So the purpose of lysing the red blood cells in chocolate agar is that kailangan natin yung mga nutrients present dun sa red blood cells kasi yun yung magpo-provide ng additional nutritional requirements for our fastidious organisms like Neisseria. Okay? So sa blood agar, hindi tutubo ang ating mga pathogenic Neisseria because the nutrients they need are still inside the red blood cells, so hindi nila ma-access yun. They're also capnophilic, meaning they need increased carbon dioxide concentration or carbon dioxide content in the atmosphere in order to optimally cultivate these organisms. That's why kapag nag-culture tayo ng ating mga possible or suspected Neisseria species, nilalagay natin siya sa candle jar. Okay, para mas facilitated yung growth nila kasi sa candle jar, nag-i-increase ang carbon dioxide concentration. The most species of Neisseria are carbohydrate fermenters. So, karamihan naman sa kanila ay nagpa-ferment ng at least one carbohydrate. But there are some species of Neisseria, yung mga opportunistic and even the non-pathogenic ones, are asaccharolytic or hindi sila nagpa-ferment ng carbohydrate, which is one useful tool in differentiating your pathogenic from non-pathogenic Neisseria. Kasi yung mga pathogenic Neisseria natin, sila yung nagpo-ferment ng carbohydrate. They grow best in culture media with blood and cholesterol, and they are non-pigmented colonies generally except for certain species like Neisseria flava, Neisseria flavescens, and Neisseria subflava. They are very sensitive to heat and drying. That's why if a patient has suspected Neisseria infections, in order to optimally facilitate the cultivation or culturing of our suspected pathogen, dapat ang inoculation natin with our primary media is through bedside inoculation. So from the patient bedside, pagkakuha ng sample, i-inoculate agad siya. Or if it's not possible, dapat gagamit tayo ng mga appropriate na transport media in order to maintain the viability of our suspected pathogen. Now, under the microscope, your Neisseria species appear gram-negative. They are diplococci, so they appear in pairs, resembling coffee bean or kidney shape. Okay? So in this image here, ayan, no? So they appear as diplococci. They also appear as gram-negative, kaya pink yung kanilang kulay. And isa to no, sa mga parang unique characteristic niya. No? So yung gram staining niya, kaya pag nakakita tayo ng gram-neg na diplococci, so ang unang papasok na agad sa isip natin na probable na pathogen ay yung Neisseria species. So they appear as coffee bean or kidney shape. On colony or on culture, the colonies are small, okay, and grayish white, opaque, convex, and glistening. So these are examples of your Neisseria colonies on chocolate agar plate. Okay, so hindi siya tumutubo again sa blood agar kasi nga kulang yung nutritional requirements doon or kulang yung nutritional content na kailangan ng ating Neisseria para tumubo siya. So they test positive for the catalase and oxidase test, and they are found in mucous membranes and respiratory and urogenital tracts. Okay. So yung ating mga Neisseria, hindi man sila lahat pathogen, no? So some of them are normal microbiota of different areas of our body, no, such as the respiratory and urogenital tracts. Okay. So balik lang tayo dito, no, sa kidney or coffee bean shape. So generally yung Neisseria natin, they're diplococci, with an exception of Neisseria elongata, Neisseria basiliformis, and Neisseria weaveri. So yung mga nabanggit ko, they are rod-shaped Neisseria. But the rest of the species belonging to the Neisseria species, so they are gram-negative diplococci. So they generally test positive for the catalase test except for Neisseria elongata and Neisseria basiliformis. Now, for the specimen collection of suspected Neisseria infections, particularly if we're suspecting of Neisseria gonorrhoeae, so swabs should be placed in transport medium such as Amy's medium with charcoal and plated within 6 hours. Kasi very sensitive to drying yung ating organism. That's why kapag ka-swab, pagkagaling sa urogenital swab or pharyngeal swabs ng ating patients, dapat ilagay agad siya sa either dun sa primary culture media na mismo or sa transport medium. Okay. So please take note that this is the most important pre-analytical factor for the detection of your Neisseria gonorrhoeae because kapag hindi natin na-collect na maayos or hindi siya na-transport na maayos sa microbiology laboratory, so we can or it may yield false negative result kasi wala na talagang tutubo kapag patay na yung organism to begin with. That's why we have to be very careful in transporting our media suspected with Neisseria infections. We don't just use any swabs. Okay? Kasi calcium alginate and cotton swabs are inhibitory to your Neisseria gonorrhoeae. That's why swabs made of dacron or rayon are recommended. Neisseria gonorrhoeae will not be recovered from sodium polyanethol sulfonate blood cultures due to its inhibitory property. So, although yung SPS natin, generally, hindi niya na-inhibit yung karamihan ng mga non-fastidious organisms natin, but for the fastidious ones, na-inhibit niya. That's why, kung suspected na septicemia with Neisseria gonorrhoeae, so hindi tayo dapat gagamit ng SPS. Yung mga culture media naman na available ngayon, so they are made of nutrient broth with resins that can remove kung mayroong mga antibiotics present na doon sa blood ng patient natin, as well as it can also inhibit the antibacterial properties of the blood. Okay. So yun na yung ginagamit natin, yung nasa bote na blood culture bottles. So, general tests for Neisseria. So, ito yung mga usual na ginagamit natin in order to differentiate pathogenic Neisseria from the non-pathogenic ones, as well as ito yung ginagamit din natin in order to differentiate your Neisseria species from Moraxella catarrhalis kasi they appear the same under the microscope. So first is carbohydrate utilization test. So like we have mentioned, your pathogenic Neisseria, they utilize at least one carbohydrate. So yung ibang mga non-pathogenic species of Neisseria, they do not utilize or ferment carbohydrates. So ginagamit natin to in order to differentiate those two types of organisms under Neisseria. So this is the standard method for the identification of Neisseria gonorrhoeae, and it detects acid production from the following sugars. So we have glucose, maltose, lactose, fructose, and sucrose. So tinitingnan natin yung fermentation patterns ng ating mga organisms with these sugars. So the culture medium is your cysteine, trypticase, or tryptone agar with 1% of each carbohydrates. Okay, so 1% glucose, maltose, lactose, fructose, and sucrose. And the positive reaction is the formation of yellow colonies or changing of your media, changing color of your media from red to yellow. Kasi di ba si phenol red, ito rin yung ginagamit natin sa mannitol salt agar. So yung original na color niya is red, and since your phenol red is an acid indicator, once acid is produced from the fermentation or utilization of any of these sugars, so it will turn the media yellow. Okay? So magbabago yung kulay niya. So again, positive reaction is yellow colonies formation or changing of the color of the media from red to yellow within 24 to 72 hours after incubation at 35 degrees Celsius. Okay, so examples are the following. So ito control, negative control. So talagang dapat hindi siya mag-change ng color. In this second tube, ang inoculate natin dito through stabbing is yung Neisseria gonorrhoeae, ATCC 1942-4. In here, Escherichia coli. So yung mga Enterobacteriaceae natin, they're also utilizing carbohydrates. That's why magpa-positive din sila. Neisseria meningitidis for this one. And your Streptococcus pneumoniae on this one. So lahat sila mag-utilize ng carbohydrate. Okay. And then we have the oxidase test, wherein we use the reagent 1% tetramethyl paraphenylenediamine dihydrochloride. So in this test, the reagent is dropped directly onto the plated colonies or an isolated colony is rubbed on a reagent disc. So may dalawang method tayo dito, no? So either yung reagent natin na ipapatak mismo dun sa colonies sa ating chocolate agar, or pwede rin na ilalagay natin, no, or i-rub natin yung mga isolated colonies dun sa ating reagent disc impregnated with your reagent. So the positive reaction is the purple color formation within 10 seconds. So dito sa pag pinatak mo mismo dun sa colonies, kailangan yung colonies magiging violet. And kapag dun sa disc method, kailangan magkakaroon ng purple coloration dun sa ating disc within 10 seconds. So that indicates a positive result in our oxidase test. And then we have the catalase test. So which, of course, na-discuss na natin, di ba? Ang positive reaction is effervescence or yung bubbling. Pag negative, so walang mangyayari. So the difference between these catalase tests and dun sa mga previous na catalase test na ginamit natin for gram-positive Cocci, dito mas mataas yung concentration natin. So about 20 to 30% hydrogen peroxide. We also have the deoxyribonuclease test or the DNase test. So nabanggit na rin or na-discuss na rin naman natin to during our lecture with gram-positive Cocci, no? So ang positive dito ay kapag nagkaroon ng clearing ng area around the colony. Kasi kapag nagpo-produce ng DNase yung ating organism, so masisira niya or ka-cleave niya tong compound na to, no, yung DNA methyl green natin. So again, the positive reaction is a formation of clear halo around the colonies after 18 to 24 hours of incubation. So this is a definitive test for Moraxella catarrhalis because your Moraxella catarrhalis produce this enzyme, while our Neisseria species, so they are negative for this one. Okay, so let's now discuss the pathogenic Neisseria, starting with Neisseria gonorrhoeae. Neisseria gonorrhoeae is also known as gonococci or gonococcus. So this is an overt pathogen, this is an overt pathogen, and humans are the only known host. So hindi siya part ng normal flora natin, and once makapasok to sa katawan natin, particularly sa mga mucous membranes, so they will start colonizing that mucous membrane and will cause disease and inflammation. Your Neisseria gonorrhoeae is the most common cause of sexually transmitted infection or STI and can be isolated from the following areas. So, pinaka-common of course is the urogenital tract, okay, kasi nga sexually transmitted siya, anorectal area, oropharynx, and even conjunctiva. Okay? So, your Neisseria gonorrhoeae is acquired or is transmitted through penetrative unprotected sex. Okay? So, kapag yung sexual partner ay may gonorrhea, okay? So, kapag meron siya nitong organism na to at nagkaroon ng sexual contact na unprotected, so pwede niyo itong maipasa, no? Kaya, common na areas na pwedeng ma-isolate ang gonococci natin ay dun sa mga areas na to. And even sa conjunctiva, if the eyes were exposed to infected reproductive fluids such as the semen. Okay. It can be also transmitted from mother to baby during delivery. So yung mga gonococcal cases natin ng mga babies, or mga children less than 1 year old, so they are usually yung vertical transmission from the mother. Okay, so ibig sabihin, nung pinanganak siya, yung mother ay infected ng Neisseria, so nakuha ng baby yung infection. Please take note that although this applies in the US, they consider gonococcal diseases or gonococcal infections in children more than 1 year old as an indication of sexual assault. Okay? Kasi imposibleng magkaroon pa ng gonococcal infection yung bata more than 1 year old na galing sa nanay. Okay? As a result of vertical transmission. Okay. So the main virulence factor of our Neisseria gonorrhoeae is the common pili. So kung maalala nyo, di ba, ang pili is it's a part of your bacterial cell that is required for attachment. Okay, so ang pinakaginagawa kasi ni Neisseria gonorrhoeae once it gains access to your mucous membranes, particularly sa urethra, okay, or sa vaginal tract. So kakapit mismo siya dun sa mucous membranes, no, through these common pili, and then magsa-start na siyang mag-cause ng inflammation and colonization. So other factors of your Neisseria gonorrhoeae include transferrin receptors. Kasi yung ating Neisseria, they need iron for growth. So that these transferrin receptors, they may it allows the bacteria to acquire iron from the transferrin, di ba? Yung transferrin kasi ito yung ating transporter ng ferric iron, di ba, sa circulation. So makakakuha sila ng iron from these transferrin with the use of their receptors. They also have different cell membrane proteins, lipo-oligosaccharide that serve as the parang toxin producing part, and then they have the IgA protease. Yung IgA class, this is a type of antibody that imbues mucosal immunity. So itong IgA, makikita to sa mga mucous membranes natin or mucosal surfaces. Now, Neisseria gonorrhoeae, since meron siyang IgA protease, so pwede niyang labanan tong protective mechanism natin, no? So it can cleave or degrade our IgA. Kaya nakakapag-colonize siya sa ating mga mucous membranes. Colonies of Neisseria gonorrhoeae on chocolate agar are small, tan-colored, and translucent and raised. And yung ating mga colonies ng Neisseria gonorrhoeae, we have different types mula types 1 to 5, and these are based on the presence of pili. Kasi hindi naman lahat ng Neisseria strains or Neisseria gonorrhoeae strains ay may pili. Okay? So yung T1 and T2 types, so sila yung virulent, okay? And sila yung may pilay. Kasi 'di ba ang most or main virulence factor natin is the common pilay. So yung mga virulent strains natin, definitely they have this. And for T3 to T5, wala silang pilay kaya avirulent sila. So let's go to the diseases caused by your gonococcal infection. Okay. So of course, ang pinaka-associated na infection caused by your Neisseria gonorrhoeae is gonorrhea. Okay, so that we will discuss this in details later. Purulent urethritis and cervicitis are manifestations of your gonorrhea. So, ayan. Anorectal infections kasi 'di ba nakukuha din yung gonococcal infection through unprotected penetrative anal sex. Kaya napupunta rin dyan yung ating gonococci kapag infected yung partner, for example. Pharyngitis, so pwede rin mapunta sa lalamunan yung ating gonococcal infections due to oral sex. Kapag sa conjunctivitis, through exposure of the eyes through infected reproductive fluids such as the seminal fluid, and of course, purulent arthritis kapag naging disseminated na or kapag kumalat na yung infection. Now, in cervicitis particularly, untreated gonococcal cervicitis may lead to sterility, so pwede siya makabaog. It can also lead to ectopic pregnancy or perihepatitis, or what we call the Fitzhugh-Curtis syndrome, or inflammation of the hepatic capsule without the involvement of the liver parenchyma. Okay? So that is particularly for females. Okay? Sa males kasi, ang common na manifestation ay yung urethritis. Okay? Nagsa-start siya sa colonization ng bacteria sa urethra. And then kapag untreated siya, pwede siyang kumalat in other areas. So pharyngitis is the chief complaint of symptomatic oropharyngeal infections. So para siyang strep throat, pero ang infecting agent natin ay of course yung Neisseria gonorrhoeae. Anorectal infections causes rectal pain and bloody stool. Kasi nga nai-inflame, nai-infect yung rectum. So pag nag-pass out ng stool yung patient, so pwedeng may kasamang blood. Right, so for gonorrhea, so it was coined from the Greek words gonos and rhoia. Okay, so gonos meaning seed and rhoia meaning flow. Kasi during the 2nd century, syempre wala pa silang idea of infectious agents. So that's why they named gonorrhea as such, because they thought first that the purulent discharge in males were actually or they thought na parang premature na ejaculation yun, yung purulent discharge. So they thought that seminal fluid yung lumalabas. Well, in fact, nana na siya. It causes acute pyogenic infection of non-ciliated columnar and transitional epithelium. And the usual sites of infection are, of course, yung mga reproductive sites natin or yung mga associated with our sexual organs such as the urethra, endocervix, anorectal area, and conjunctiva if na-expose yung mata. Right. So these are ito yung mga examples, or yung manifestations, examples of manifestations of gonorrhea. Ayan. So for urethral discharge, penile urethral discharge, so yung mga tao nung 2nd century, they thought first that this purulent discharge is actually semen. So makikita naman sa difference, kasi ang semen is grayish white in color and this one is yellowish. So magkaiba yung kanilang kulay. At the same time, if a patient is suffering from this one, so aside from observing urethral discharges, yung purulent urethral discharge, masakit din yan, it causes, or it is described as with having burning sensation while urination, so mahapde. And for cervicitis, ito yung cervix, no, yung opening ng, ng uterus. Okay? So ayan, so nakikita nyo merong ring purulent discharges, so yung nana. Okay? So this is caused by your Neisseria gonorrhoeae. And for the pharyngitis, so ito yung oropharynx ng isang patient. So makikita nyo yung mga whitish spots. So they are actually purulent discharges, mga nana yan that are caused by your Neisseria gonorrhoeae dun sa lalamunan. And of course, conjunctivitis, it can cause purulent discharges sa ating mata kapag nagkaroon ng gonorrhea sa mata. Ay, pag na-infect yung conjunctivitis. By the way, ano ba sa Tagalog ang gonorrhea? So it is known as tulo. Okay? So siya yung pinaka-common na STD or STI among the other sexually transmitted infections. Siya yung pinaka-common. So signs and symptoms of the disease include purulent urethral discharge, such as these and these. It can cause lower abdominal pain kasi pwedeng kapag nagdi-disseminate na, no, yung infection. So pwede na siyang kakalat. So from the urethra or the cervix, pwede na siyang umakyat into other areas. So yung lower abdominal pain, particularly mararamdaman siya ng women. Okay? And it may cause also vaginal bleeding kasi meron siyang infection. Right? However, in most cases, class, ang gonorrhea ay asymptomatic among females. Okay? Kasi 'di ba sa females, normal talaga naman na may mga discharges. So kapag hindi ka aware dun sa mga normal discharges ng vagina, so pwede mong overlook, no, kung meron ka ng gonorrhea or wala. Sa males, madali siyang ma-detect kasi nga it is characterized by, again, purulent urethral discharge at the same time burning sensation while urinating. Okay? So this is for gonorrhea. We also have a specific type of gonorrhea called your ophthalmia neonatorum, which affects particularly yung mga newborns natin. Kasi 'di ba sabi natin nga ang gonorrhea ay makukuha din ng mga babies kapag or during normal delivery from an infected mother. So this is a gonococcal eye infection acquired as the baby passes through the infected birth canal. Kasi 'di ba ang nai-infect ay yung cervix, doon dadaan yung baby kapag or during childbirth. So it may lead to corneal ulceration and eventually blindness if left untreated. That's why it's a standard practice during delivery, standard practice na kapag lumabas na yung baby, nilalagyan ng erythromycin ointment yung mata ng baby, regardless kung ano yung gonorrhea status ng mother. So para maiwasan, no, or para ma-prevent na agad kung meron mang Neisseria gonorrhoeae na napunta dun sa mata ng ating baby. But before erythromycin class, ang ginagamit during the old times ay yung 1% silver nitrate. So hindi na natin ginagamit yun ngayon kasi nga heavy metal siya. So accumulation of that heavy metal may lead to health effects. Okay? So those are the different diseases caused by your Neisseria gonorrhoeae. So let's now proceed with the laboratory identification or diagnosis of gonococcal infections. So we have, of course, the gram staining on culture as a staple in the clinical microbiology lab. In the immunology and serology laboratory, we can also perform serological tests that detects specific antigens of your bacteria. We can also perform molecular tests, particularly for those if we are looking for yung mga antimicrobial genes. Okay? And then, of course, the standard antimicrobial susceptibility in differentiating isolates. For gram staining, of course, the specimens that we use for direct microscopic examination as well as for culture, so purulent discharge from the urethra, cervix, prostate, rectal mucosa, yung mga eye swabs, and joint fluids in the case of purulent arthritis. So ito yung mga ginagamit natin for direct microscopic examination. Please take note that smears from throat swabs should not be done due to presence of commensal Neisseria species. So dito lang tayo nagpa-perform ng direct microscopic examination. Sa throat, hindi kasi meron tayong mga commensal or mga normal na Neisseria sa lalamunan natin. So pag throat swabs, diretso na siya sa ah, pag-inoculate sa ating primary plates. So under the microscope, they appear, or you can observe intracellular gram-negative diplococci. So they appear in pairs. They also are gram-negative, so pink ang kulay. So with the characteristic coffee bean or kidney bean shape. So isa sa mga hanapin nyo, class, if you received a purulent discharge sample suspected for Neisseria, ang una nyo expected na makikita are, of course, yung abundant neutrophils. Okay, so these are all neutrophils responding to the infection. And to confirm, ayan, makikita nyo na may mga diplococci sa ating intracellular diplococci dun sa ating neutrophils. Ito yung mga engulfed na diplococci. So they appear in pairs, okay, and have this characteristic coffee bean or kidney bean shape. Ayan. So this is a classic example of a direct microscopic examination from a purulent discharge caused by gonococcal infection. So presence of more than 5 PMNs per immersion field without bacteria is suggestive of non-gonococcal urethritis. So ang unang hinahanap nga natin, 'di ba, is yung mga polymorphonuclear cells or yung mga neutrophils. At ang susunod yung hahanapin ay yung mga intracellular diplococci. So kapag nakita nyo to, so that is suggestive of gonorrhea. However, kapag nakakita kayo ng abundant na white blood cells pero walang bacteria, so that could be an indication of non-gonococcal urethritis, particularly caused by chlamydia trachomatis. Si chlamydia, it's also a sexually transmitted infection, but they are not susceptible or they are not detectable through gram staining. Okay? Kasi intracellular parasites ang ating chlamydia. Okay. For culture, okay, so we can use chocolate agar as our base. So again, bakit siya tinawag na chocolate agar? It's not because it's made of chocolate, but it's made of lysed blood. Okay, so para siyang dinuguan. Kasi di ba sa dinuguan, oxidize yung ating blood kaya nagiging brown. So ganun din siya. And the purpose of lysing our red blood cells upon addition in our melted agar in the preparation of CA is that para makuha natin yung mga additional requirements or additional nutrients present inside the red blood cells to facilitate the growth of our fastidious organisms such as Neisseria. We can also use other culture media that are also based on chocolate agar, such as the Thayer-Martin agar, modified Thayer-Martin medium, Martin Lewis, and we can also use New York City medium and G-Select. Okay? So itong apat na to, sila yung based dun sa lysed blood. Okay? Kaya ang kulay nilang apat ay brown. And for NYC and G-Select, they are clear culture media for Neisseria. Now, like we have mentioned, yung Neisseria gonorrhoeae will not grow on blood agar because sa blood agar, 'di ba, yung mga nutrients na kailangan ng ating bacteria ay nasa loob pa ng red blood cells. That's why hindi sila tutubo basta-basta. So in order to facilitate their growth, dapat malyse yung ating red blood cells. Kaya gumagamit tayo ng chocolate agar or any media that are based on chocolate agar or kung meron mga supplements. Bedside inoculation is recommended because of its susceptibility to drying. So napaka-sensitive ng ating Neisseria gonorrhoeae, and kapag hindi siya na-transport na maayos to the laboratory, so wala na, patay na yung organism natin, which can lead to false negative results kasi wala talagang tutubo sa ating media kung patay na yung organism. Immediate incubation of at 35 degrees Celsius to 37 degrees Celsius for 72 hours with increased carbon dioxide is required to achieve optimal growth because they are capnophilic. Okay. So for the culture media, again, so these are based on your chocolate agar. So starting with Thayer-Martin agar. So it's a chocolate agar with enrichment supplement with the brand name Isovital X, and certain antibiotics in order to inhibit the growth of contaminating organisms. So yung mga ating antibiotics found in your Thayer-Martin agar includes your vancomycin, your colistin, and nystatin. So si vancomycin, it inhibits your gram-positive bacteria. Your colistin inhibits gram-negative but not Neisseria. Okay, and your nystatin it inhibits fungal contaminants, particularly mga yeast. For modified Thayer-Martin or the MTM medium, it includes all the components of your Thayer-Martin agar with an exception of trimethoprim na dagdagan siya. Okay? So in addition to trimethoprim, now this is to inhibit swarming organisms such as proteus, di ba? And then we have the Martin-Lewis medium, which contains all the contents of the modified Thayer-Martin medium, but with increased vancomycin concentration, and yung nystatin natin ay pinalitan ng anisomycin. So that's the Martin-Lewis medium. And then we have the New York City medium or the NYC medium. So this is a transparent medium with lysed horse blood, horse plasma, and yeast dialysate. It has the same antimicrobials as the modified Thayer-Martin medium, except anisomycin may be replaced by amphotericin B. Kasi in some preparations, anisomycin pa rin yung isa sa mga antibiotic, but in some, amphotericin B na. And then we have the G-Select medium, which have the same which has the same antimicrobials as NYC, okay, with the addition of lincomycin. So this lincomycin also works against gram-positive organisms. And then for transport media, we have the following. We have the Transgrow, the Clary Blair, Ames, and even the Gembec medium. So ginagamit natin ang transport media if bedside inoculation with our specimen is not possible. And then for the serological tests, okay, so of course we can have the coagglutination test, which detects specific gonococcal antigens with the use of reagent antibodies. Okay? So for fluorescent antibody test, parang ganun din siya yung principle. We are detecting gonococcal antigens, particularly the principal outer membrane protein of your gonococci. However, the difference in this setup is that instead of observing agglutination such as in here, ang ino-observe natin ay yung fluorescence. Okay? So it's a type of labeled immunoassay. So in immunology, we will discuss further yung concepts ng fluorescent antibody tests. But the similarity between these two, so we are detecting specific gonococcal antigens or yung mga markers that are present on the surface of your Neisseria gonorrhoeae, which are specific for that bacteria only. Kaya kapag naka-observe tayo ng positive reactions, so most probably the isolate is Neisseria gonorrhoeae. We can also perform molecular tests such as nucleic acid amplification test or yung NAT. We also have the chemiluminescent nucleic acid probe, among others. So hindi limited dito yung ating mga molecular tests. For the NAT, ito yung isa sa mga pinaka-common. Okay, so it detects gonococcal nucleic acids directly from specimens. And our specimens, of course, yung mga endocervical and urethral swabs and even urine. So ginagamit natin itong NAT kapag nagde-detect tayo ng mga STDs or STIs. So the advantage in some setups of nucleic acid amplification test, so it runs a series of identification and not just for gonococci, it can also detect other agents of urethritis such as chlamydia. For the chemiluminescent nucleic acid probe, it's a rapid test for detecting gonococcal ribosomal RNA in genital or conjunctival specimens, and it provides results within 1 to 2 hours compared to your conventional nucleic acid amplification, which will take about 6 hours. Ganun. So, ganun din yung specimens natin, endocervical, urethrals, conjunctival swabs. So, yung mga nabanggit nating specimens kanina, so pwede yon. Okay. So dito rin pala sa nucleic acid amplification test, it's not just limited to urethral samples or yung urogenital samples, basta lahat ng samples na suspected of carrying the Neisseria gonorrhoeae, so pwede natin gamitin dito. And of course, we have the antimicrobial susceptibility test. The preferred medium is the gonococcal or the GC agar, and the preferred method is disc diffusion or agar diffusion. And we have an alternative method, of course, we have the epsilometer test. And ang mga tinatest natin for antimicrobial susceptibility for Neisseria gonorrhoeae are yung mga extended spectrum cephalosporins natin and mga quinolones such as yung mga levoflox, ciproflox, gatiflox, mga ganyan. Now, cephalosporins are the treatment of choice for gonorrhea due to widespread penicillin resistance. Okay? So, it's also a problem now kasi yung ating Neisseria gonorrhoeae, nagkakaroon na ng emergence ng multi-drug resistant. Kaya, there are cases now of gonorrhea that are untreatable. Hindi na siya gumagaling. So another pathogenic Neisseria organism is your Neisseria meningitidis. Neisseria meningitidis is also known as meningococcus or meningococci. This is the causative agent of meningococcemia and the leading cause of fatal bacterial meningitis. And it may be found as a commensal of the upper respiratory tract of some individuals, but your Neisseria meningitidis is never a part of your normal flora. Okay? So, yung mga carriers natin, so, di ba pag sinabi natin carriers, sila yung mga tao that do not present the illness or they do not manifest the disease. However, they can transmit the infectious agent to another individuals. And sa ibang individuals, if they're susceptible enough, doon pwede mag-manifest yung disease and it may even become contagious from there on. Your Neisseria meningitidis organism is a glucose and maltose fermenter. It also requires iron for growth, and it's also producing beta-lactamase. On colonies on BA, it appears bluish-gray and mucoid, and they appear small, tan, and mucoid on chocolate agar. So isa sa mga difference ni Neisseria meningitidis and Neisseria gonorrhoeae is that your Neisseria meningitidis may grow on your blood agar while your Neisseria gonorrhoeae, so very fastidious siya, kaya sa chocolate agar plate lang siya or sa mga media na may supplements lang siya tumutubo. The virulence factor of your Neisseria meningitidis includes the pili and also the polysaccharide capsule, so it's an encapsulated organism. It also has IgA protease that degrades or that cleaves the IgA antibodies in the mucosal surfaces. They also have cellular membrane proteins such as 4A and 4B, and it also has the lipo-oligosaccharide endotoxin. Ang Neisseria meningitidis class, meron tayong total na 12 serogroups, but the most significant serogroups are A, B, C, Y, and W135. Now, isa sa mga pinaka malalang sakit na kinukos ni Neisseria meningitidis, of course, is yung meningococcemia. So, meningococcemia is defined as the presence of Neisseria meningitidis in the bloodstream and can occur with or without meningitis. Kasi ang pinaka parang associated disease talaga ni Neisseria meningitidis ay yung meningococcemia and meningitis. Meningococcemia is spread via droplet transmission and it's very contagious. It's very transmissible, no? Kaya yung mga suspected natin na patients with meningococcemia, so they are immediately isolated kasi pwede silang makahawa agad. And your meningococcemia, it's a fatal infection with the following signs and symptoms: frontal headache, stiff neck, pero ang pinaka characteristic na manifestation ng meningococcemia is the fever with petechial rashes. Ito. So these rashes, they are not actually rashes but they are actually subcutaneous bleeding. Okay, so nagbe-bleed na yung patient natin. Okay. So these are mga petechia. And in fulminant cases of meningococcemia, the bacteria can spread rapidly, causing disseminated intravascular coagulation and septic shock or adrenal hemorrhage, or what we call the Waterhouse-Friderichsen syndrome. So very fatal no ang meningococcemia, kaya emergency 'yan. However, yun nga, very contagious din siya, kaya uh, they are treated with great caution, no. Okay. So this is an example of adrenal glands with hemorrhages. So kung makikita niyo yung uh, mga dark red spots, so they are actually bleeding. Okay, so nag-bleed yung adrenal gland, leading to, of course, a shock and certain hormonal imbalances. Kasi di ba, your adrenals, they are one of your endocrine glands. So pag nasira siya, so it can lead to several hormonal imbalance as well. And yun nga, again, your fulminant cases can spread rapidly, causes DIC and septic shock, which can lead to death within 12 to 48 hours post-onset of symptoms. So again, it's very fatal. That's why kaya 'di ba kung maalala nyo sa CPH natin and sa epidemiology, meningococcemia is one of the uh, notifiable diseases. Okay? Kasi mabilis siyang kumalat, at the same time it's very fatal. So for the laboratory identification or diagnosis of meningococcal infection, it includes, of course, your gram staining and culture. We have the oxidase test, antimicrobial susceptibility, and serological test. However, class, itong mga uh, test na to, it's not really that useful, no, in the identification process of, or in the immediate diagnosis rather, of the disease, kasi nga napakabilis ng progression niya. So once mag-appear kasi yung symptoms, particularly the rashes, if untreated, if left untreated or if poorly managed, it can lead to death within 12 to 48 hours. Kaya usually itong mga laboratory diagnostic methods natin are for confirmatory purposes na lang. So kapag may impression na yung physician na it could probably be meningococcal infection, they will proceed immediately with treatment. Pero syempre, gagawin pa rin natin itong mga laboratory test, pero hindi na siya yung talagang useful, no, for um, diagnosis, kasi they will treat right away the patient with suspected meningococcal infection until such time na ma-rule out ang meningococcemia. And of course, yung laboratory diagnosis natin is very helpful in reporting these diseases to health authorities. For gram staining and culture, so they also appear gram-negative intracellular and extracellular diplococci. So in the case of meningococcal meningitis, so ang una nyo makikita kapag nag-perform kayo ng CSF microscopy or yung CSF fluid cell count, so makikita nyo yung ating mga abundant white blood cells, particularly yung mga neutrophils. Kasi ang normal na CSF natin, class, almost non-neutrophils. Okay? So kapag abundant ang neutrophils, that indicates bacterial infection. And kapag nakakita kayo ng mga diplococci, intracellular and extracellular diplococci like these, okay, dun sa ating CSF sample, so that is indicative of meningococcal meningitis. So or yung culture media natin may include blood agar, chocolate agar, and Thayer-Martin agar. So nasopharyngeal swabs should be plated immediately in Gembeck system or submitted on swabs that are placed in charcoal transport media in order to preserve the viability of our organism. SPS should not concentration in blood culture broths should not exceed 0.025% because above this, it may inhibit now your meningococci if we are using blood as a sample. For antimicrobial susceptibility, the Clinical and Laboratory Standards Institute recommends either broth dilution or agar dilution MIC test with cation supplemented MH broth and laked horse blood, or MHA with 5% sheep's blood. So for example, mukha siyang blood agar, but ang base nito ay Mueller-Hinton. Okay, so ito yung dapat ginagamit natin for antimicrobial susceptibility of Neisseria meningitidis. And for the treatment option for uh, meningococcal infections, of course, the choice of drug is number one, penicillin. But for penicillin, particularly for meningococcal meningitis, but for meningococcemia, it's a type of septicemia, di ba? So, ang binibigay natin agad is cephalosporins, okay, like we have mentioned previously. Other tests include mga serological tests such as latex agglutination and counter-immunoelectrophoresis. These are just among the different serological tests that we may use in order to facilitate detection of our uh, pathogen. We can also perform several biochemical tests such as oxidase and gamma-glutamyl aminopeptidase test. Okay, so these are just some of the different methods that we can use in order to detect our Neisseria meningitidis. Okay? Now, proceeding with the non-pathogenic Neisseria species, ito, hindi sila masyadong concern or hindi sila masyadong parang clinically significant. However, they can still cause opportunistic infections. Okay? Pero the diseases that they cause are not as severe as gonorrhea and meningococcemia. So examples of the non-pathogenic Neisseria species are the following. So we have Neisseria cinerea. So the colonies on CA appear similar to your T3 colonies on of Neisseria gonorrhoeae. So di ba yung T3 and T5 natin, or T3 to T5, they are the avirulent strain. So yung wala silang pilay. Colistin susceptibility differentiates Neisseria cinerea from Neisseria gonorrhoeae. Neisseria flavescens, it's a saccharolytic, it's also pigmented colonies. So yung sa colonial morphology pa lang, makikita nyo na yung difference, no? So mas-separate nyo na siya from the pathogenic ones. It is capable of growing on blood agar and chocolate agar plate at 22 degrees Celsius. Next is Neisseria lactamica. So it is the only lactose-utilizing Neisseria, thus the name lactamica. It's isolated from meningococcal carrier surveys, commonly from children 1 to 2 years old and rarely from adults. We also have Neisseria mucosa. These are usually isolated from the nasopharynx of children and young adults. They grow on Na at 35 degrees Celsius and colonies appear large and very mucoid. Neisseria sicca. These are Neisseria species that appear dry, wrinkled, adherent, or with breadcrumb-like appearance of their colonies on nutrient agar. For Neisseria elongata and Neisseria basiliformis, so they are rod-shaped. So sila isa sa mga exceptions, no, ng ating parang general rule na ang Neisseria ay gram-negative cocci. Kasi si Neisseria elongata and Neisseria basiliformis, they are gram-negative bacilli. So they are weakly positive to negative or negative to catalase test, and they are commensals of the upper respiratory tract but are considered opportunistic pathogens. So in immunocompromised individuals, so they may cause infections. We also have your Neisseria polysaccharia, which produce large extracellular polysaccharide when grown on media containing 1 or 5% sucrose. And it can be confused with Neisseria meningitidis and is differentiated by the ability to grow on nutrient agar at 35 degrees Celsius and polysaccharide production from 1 to 5% sucrose. We also have your Neisseria subflava, which is also a pigmented Neisseria. So it's less yellow, kaya siya subflava. Flavescens kasi meaning yellow. So generally non-pathogenic but have been implicated with serious infections, particularly dun sa mga immunocompromised. And clinical description of infections may resemble Neisseria meningitidis. And then lastly, we have the Neisseria weaveri. It's also a rod-shaped Neisseria species that tests positive for catalase but does not produce any from or any acid from the carbohydrates used to identify your Neisseria species. So it's also a saccharolytic. It reduces nitrite to gas. It's weakly phenylalanine deaminase positive and is sensitive to penicillin. So again, ang pinaka-purpose kung bakit natin dinidiscuss yung mga non-pathogenic Neisseria species, it's because kailangan alam natin kung paano i-differentiate ang mga organisms na to from the pathogenic ones kasi it might cause confusion during the identification process. Okay? So, pwedeng yung na-isolate nyo pala ay contaminant lang pero ni-report niya agad as meningococcinia. No? So, mahirap yon. So, dapat alam nyo talaga yung mga nitty-gritty nung ating differentiation steps in order to correctly identify our organisms. So next we have the Moraxella or the Branhamella catarrhalis. So Branhamella yung kanyang former na genus, so it's now known as the Moraxella catarrhalis. So it's the commonly isolated species of Moraxella and belongs to the Moraxellaceae family. It's an indigenous microbiota of the oropharynx and an opportunistic pathogen of the upper respiratory tract. And it's a common cause of otitis media or yung ating middle ear infection and sinusitis in children. It resembles Neisseria species on microscopic examination. They appear also as gram-negative cocci. That's why it's very important that we differentiate Moraxella from pathogenic Neisseria through biochemical tests. They're also non-motile. They are fastidious, they are beta-lactamase producer, and they are encapsulated and with pili. So they can be easily misidentified as Neisseria if not kung kapag hindi natin ginawa yung mga proper identification steps. So Moraxella catarrhalis are small, gram-negative cocci that tend to grow in pairs from end to end with the flattened adjacent sides. Okay? Although in most cases, kapag sa microscopy naman, hindi mo siya basta-basta makikita, no? Kasi parang it really resembles yung Neisseria bacterial cells. Their colonies on BA are smooth, opaque, grayish-white, and have a hockey puck or wagon wheel appearance. So isa sa mga difference no of Moraxella catarrhalis and your pathogenic Neisseria is that it is capable of growing in your blood agar. Kasi si Neisseria gonorrhoeae, sa chocolate agar lang siya, pero si Neisseria meningitidis, no, tumutubo rin siya sa blood agar plate. That's why for proper identification, hindi lang mga laboratory test ang ginagamit natin. Yung proper identification ng infectious disease also relies with the proper patient history and physical assessment ng physician. So, ang colonies ng ating Moraxella catarrhalis are characterized or are described as hockey puck or wagon wheel. So, yung hockey puck, siya yung parang, di ba yung, kung familiar kayo sa game na hockey, di ba siya yung parang, ball, di ba yung pinagpapasapasahan na disc dun sa floor. So, kaya siya tinawag na hockey puck appearance kasi kapag yung colonies natin ay ginalaw mo using an inoculating loop, so gagalaw yung buong colonies, no? So magmo-move siya para siyang yung hockey puck nga across dun sa floor. Kasi yung mga ibang colonies ng mga bacteria, kapag ginalaw mo yung colony niya, masisira, magdi-disintegrate. Siya kapag minove mo, gagalaw yung buong or magmo-move yung buong culture, buong colony. Okay? Yung older colonies natin may have the wagon wheel appearance. For the laboratory diagnosis of Moraxella catarrhalis, so it includes of course your gram staining and culture. Pero yun nga, they appear the same with your Neisseria species. They can grow on blood agar and chocolate agar at 22 degrees Celsius and on nutrient agar at 35 degrees Celsius. They are inhibited in a gonococcal media by colistin. So kapag ininoculate natin yung specimen containing Moraxella catarrhalis dun sa mga gonococcal media natin such as yung Thayer Martin agar and the likes, so hindi siya tutubo kasi nai-inhibit siya ng colistin. For catalase and oxidase test, they also test positive, similar to your Neisseria species. Carbohydrate utilization, your Moraxella catarrhalis does not utilize any sugar in your cysteine trypticase or tryptone agar. Kaya isa to sa mga ginagamit nating pang differentiate biochemically speaking ng Moraxella catarrhalis and your pathogenic Neisseria. Kasi yung mga pathogenic Neisseria natin, they are utilizers or fermenters of carbohydrates. And one of the definitive tests for your DNA or for your Moraxella catarrhalis is your DNA or the deoxyribonuclease. Kasi ang ating Moraxella catarrhalis is positive for this one, while the pathogenic Neisseria are negative. Another definitive test is the butyrate esterase test, wherein a loop full of organism is placed onto a pre-moistened disc with the substrate, okay, which is yung butyrate. And then, ito nga, yung bromochloroindolyl butyrate, okay, so the butyrate enzyme. If the organism, such as Moraxella catarrhalis, produces the enzyme butyrate esterase, so yung ating disc ay magpo-form ng blue-green color, okay, such as this one. So yung actual size ng ating disc, para siyang yung antibiotic disc lang, no? So ito parang in-enlarge lang. And so blue-green appearance or blue-green formation on the disc or the reagent disc. Now, to summarize the different biochemical tests for the identification of your Neisseria and Moraxella catarrhalis, so these are the following. So for catalase test, magpa-positive ay yung ating mga pathogenic Neisseria but not for Moraxella. Growth on gonococcal medium or yung mga Neisseria medium natin such as modified Thayer-Martin, Martin-Lewis, and even your New York City medium. So mag-grow ang ating Neisseria gonorrhoeae and your Neisseria meningitidis, while yung Moraxella catarrhalis may be variable. Pwedeng oo, pwedeng hindi, but usually hindi. Growth on blood agar or chocolate agar at 22 degrees Celsius. So yung Moraxella catarrhalis lang yung pwedeng tumubo nun, although variable din. But for your pathogenic Neisseria, so hindi. For nutrient agar at 35 degrees Celsius, ang positive dyan ay si Moraxella catarrhalis but never on our pathogenic Neisseria. Acid production, of course, si Moraxella catarrhalis, it's a saccharolytic, so it won't produce any acid from these sugars. Yung ating Neisseria gonorrhoeae ay positive lang for glucose, while yung ating Neisseria meningitidis is positive for glucose and maltose. So isa to sa mga pwedeng gamitin pang differentiate ng ating Neisseria gonorrhoeae and meningitidis. But for the acid production from lactose, sucrose, and fructose, negative ang ating Neisseria gonorrhoeae, Neisseria meningitidis, and Moraxella catarrhalis. But for the other non-pathogenic Neisseria species, they may test positive on these. Kaya pwede natin gamitin to as a differentiating step for Neisseria, yung mga pathogenic and non-pathogenic Neisseria. Deoxyribonuclease test and your butyrate esterase test, they are definitive test for Moraxella catarrhalis as well as your nitrate reduction. Ayan. So, siya lang yung magpa-positive while your pathogenic Neisseria, they will test negative for this. So these are the different biochemical tests for the identification of Neisseria species and Moraxella catarrhalis. So that ends my lecture on gram-negative cocci. These are my references. Thank you very much for listening.